Today I will be talking about DNA replication. DNA is made up of two strands twisted around each other in a double helix. The strands are made of 4 nitrogenous bases, A, the green one, to T, the red one, and C, the yellow one, to G, the blue one. This means that they are complementary. A will always go to T and C will always go to G. The strand is 5' to 3'. These two strands run in opposite directions. So on the top it will be 5' to 3' going to the right, and on the bottom 5' to 3' going to the left. This determines how each strand is replicated. The first step is to separate the two strands. The enzyme helicase separates these two strands. These separated strands create the possibility of forming a new strand of DNA. The enzyme primase starts the process by making a small piece of RNA called a primer, which is the starting point of a new strand of DNA. The enzyme DNA polymerase binds to the primer and makes a new strand of DNA. It only adds DNA bases in one direction, which is 5' to 3' in this direction. And there's two strands that form. The leading strand is made continuously because it only adds bases one by one. However, the lagging strand cannot be made in this continuous way because it runs in the opposite direction, 3' to 5'. It is only made in small chunks called Okazari fragments. Each fragment is started with an RNA primer, and then DNA polymerase fills the space from 5' to 3'. The enzyme exonuclease removes RNA primers from both strands. DNA polymerase then fills in the gaps with DNA. The enzyme DNA ligase seals up fragments of DNA to form continuous double strand.